So around 10:40 I started class till 10:20. Sorry, 11:25 I have taken the class.
No, sir. I log in, but it is not showing the details.
No, I don't know, but it was scheduled, no, Friday, Saturday, Sunday.
Yes, sir.
Yeah,
Yesterday I log in the class, but the link is not there. Video classes are not set schedule only they are showing.
even live streaming also started yesterday. Even now is also now also it's live stream going on. Okay, no issue. Fast is fast. We can't do anything about it. So we have today and tomorrow 2 classes, no.
Yes,
So
sir.
we'll go to this block one developmental biology unit one, right?
Sir, in yesterday's class, what you have covered?
Yeah, I have covered the plants in primary healthcare. After that, traditional medicine versus modern medicine, pharmacognosy, and plant food drugs. So that block four over, and ninth unit of block three is over. So today this will finish block one developmental biology, and tomorrow we'll finish block two embryology and paleontology. Don't worry, I have made slides. So you do one thing, just share your emails, or else you tell me your email. I'll just forward this PPTs to you.
Okay, sir. Okay.
Yeah, you can write your email in the chat box. Okay, right. I will start with this one, developmental biology. So flowering plants, okay, angiosperms basically. Flowering plants produce haploid spores. So in there will be two types of cell divisions, as if you are aware of. So mitotic divisions will be there, meiosis divisions will be there. So mitotic divisions means they are occurring in the non-germinal cells like somatic cells. Okay, that will be helpful in development of a plant growth and development. They'll divide and they'll form new cells, so that new tissues will be formed. So new organs will be formed and plant will be developed. So whereas in meiosis conditions, so in meiosis and in meiosis two, so we'll get haploid germinal cells kind of. So like in male gametes, we'll get male gametophytes like pollen grains will get. In if you come to female reproductive system, we'll get a mega spores. We can say ovule kind of things will be produced in this meiosis two. Okay, so microspores are produced within the anther, whereas the megaspores are produced within the ovule. Right. So from the ovule, we'll get the megaspores, and from the anther, we'll get the microspores. They're nothing but pollen grains. Okay. A successful angiosperm pollen grain, that is gametophyte, containing the male gametes, are transported to the stigma. Right. If you could take this one, this one. Yeah. If it is a female reproductive female flower, if you can say. So this is stigma, right? This is a stigma. Can you see this cursor?
No, sir.
Cursor is moving. No? Cursor, I can see on this green part. You can see green image. Cursor is
Image,
moving. No?
see, sir, but cursor is not there.
See, now you can see on the green stigma place it is moving. Because I'm also logging from mobile, I can see this one. Okay, so actually you can see the labels. No, that the top one is stigma, and this long tube-like structure is known as style, and this is ovary. Within the ovary, ovules will be there. So within the ovule, you can see nucleus will be there. Two polar bodies will be there, and egg cells will be there. So all togetherly, we can say female gamete is one. Okay. So what they are telling here, a successful angiosperm pollen grain, that is gametophyte, containing the male gametes, are transported onto the stigma. So stigma means this one, this starting portion of the female reproductive organ. Okay. So where it is gametes and pollen tube grows down to the style. Okay, this is style. So this part is style. So the pollen grains will touch here. After touching here, so they'll form a long tube-like structure. Okay, pollen tube will be formed. Okay. So Each male gametophyte contains two nucleus will be there. So it's two male so male gametes travel down to the tube where the gametophytes containing the female gametes are held within the carpel. So one male gamete fuses with the ovule to produce embryo, and the other one with the polar nucleus to produce endosperm. Okay. So here nucleus will be there, no? So one nucleus of the male gamete will fuses with the nucleus to form embryo. Embryo means seed, we can say in future. Okay. So another nucleus of the male gamete will fuses with the polar nucleus. So here two polar bodies are there, no? With that, see if it is having two males, one male gamete will come fuses, it will form 3N cells. So 3N basically it will be endosperm. It is a food material for the seed and the embryo. Okay. Microsporogenesis is the formation of microspores inside a microsporangium or a pollen sac. So pollen sac means this is, okay. If you take these are the pollen grains, this thicker structure is there, no? That is the pollen sac. Okay. By mitotic and reduction division, the formation of pollen grains from the microspore is termed as microgametogenesis. So that is microsporogenesis or microgametogenesis is the formation of pollen grains within the microsporangium, that is nothing but pollen sac. Okay. Formation of microspores. The developing anther consists of two types of cells, reproductive and non-reproductive cells. We discussed now reproductive cells and stomatic cells. Those are non-reproductive cells. The diploid cells are the reproductive cells in the sporogenous tissue of microsporangium called meiocytic. Okay. The diploid cells are reproductive cells in the sporogenous tissue of microsporangium called meiocytic or pollen mother cells divide by meiosis to produce four haploid microspores. I'll show you the next thing is the first reduction division gives two nuclei, and the second division finally gives a tetrad of microspores. Means four will be there. The non-reproductive cells from the anther tissue layer, including epidermal, tapetal, and the cortical layers. So the tapetal layer functions as a nutritive tissue for the developing anther. So I'll show this one here. Yeah. So this is a okay. So this is a transfer section of a pollen grain. So see here you can see, no? So you can see stigma here, right? Yeah, stigma is there, anther is there, ovary is there. This is a complete plant, we can say. Okay. Here, if you can take this cross section of this pollen tube something, so there will be okay. This anther part, if you can take the cross section, you can see the this kind of structure. Okay. So this anther will be in a 2N diploid cell we were talking about. So this is diploid cell. After meiosis divisions, the microspores means they are haploid in nature. Okay. So haploid nucleus will go and fuse with the what ovule. So ovary inside the ovule. Okay. So these are the pollen grains. So this is what generation of microspores, nothing but pollen grains from the pollen sac is known as the microsporogenesis. Okay, this one. So here, this is a megaspore. So size-wise bigger, the female reproductive organ will be bigger in size. So that's why it is called as megaspore, and pollen grains will be small in size. That's why they called as microspores. Okay, that is, it will be in twin state. After meiosis, so it will form a four-cell structure. So this is megaspore. After that, it will go to mitotic division and it will form an egg cell. Okay, so this formation of egg cell from the ovary is known as megasporogenesis, and formation of pollen grains from the anther is known as microsporogenesis. Here you can see this is a single stamen. So, and this is the anther. So this is the cross section. Okay. So this will be like anther, filament, together known as stamen. Okay. So if you take the cross section of this stamen, you can see the pollen sac where pollen grains will be there. So when it get matures, no, it will open up like this, and from that the pollen grains will comes out. Okay. So they may be travel through air, they may be traveled by birds or butterflies. They'll go and sit here. Okay. When they are placed here, so there will be formation of pollen tube will be there. From that nucleus will be there from this pollen grains. They'll transfer into the ovary. After traveling to the ovary, they'll fuse. One will fuse with nucleus of the egg, and that will forms the embryo. Another one fuses with the polar bodies, that will form the endosperm. So this is the bigger image of this anther cross section. So you can see this is the pollen sac. Within the pollen sac, you can see the pollen grains. This red color ones are the pollen grains. Okay, so this parts. So this middle portion will be called a connective, and this outer layer will be there. No, that is epidermis. The next to the epidermis, there will be endothelium. After that, there will be middle layer, and after that there will be tapetum. Inside the tapetum, you can see the sporogenous tissue. So sporogenous tissue, which will give you the microspores. Okay. See, when it is get matures, no, so it will breaks from here. So that is known as stomium. From that, this dispersal of pollen grains will occur. So these are pollen grains. So this is a pollen sac. Okay. From that, this pollen grain dispersal will occur. So you can see that zoomed or magnified image. So this is the outer layer is epidermis. Next to the epidermis, there will be endothelium. After that, there will be middle layer. After that, there will be a tapetum layer. So within the tapetum layer, that microspore mother cells will be there. They are nothing but sporogenous tissue. Okay. So from the first sporogenous tissue, you can see the formation of pollen grains. So this is the real image of the microspore. So these are the tapetum cells. So within that, pollen grains are there. So we were discussing, no? So meiotic division. So meiosis 1, there will be a single cell. After that, after meiosis 2, there will be formation. After meiosis 1, there will be a with the single nucleus, 2-cell formation will be there. After meiosis 2, there will be 4-cell formation will be there. So they'll be forming into the 4. So there will be a wall formation will be there. So you can see this one, no? This wall secretion will be there, so that they will be separated as a single pollen grain. So these are the microspores. Okay. So there will be a division. So the division is, so this is a generative cell, and this is generative cell wall formation is there. Okay. So this is vegetative cell. Vegetative cell will help in the nourishment of this generative cell. Generative cells will help to form a pollen grain. So here you can see. This is a transfer section of anther. Okay. So you can see this structure, no? So after maturing, they'll come out. What pollen grains will come out. So there will be two types of cell divisions will be there. One is successive division, and another one is simultaneous division will be there. So if you do a successive type of cell division, okay. So it will form two. So this one is DNA. You can say the separation, no? So anaphase, there will be a separation of chromosomes. So after this, there will be formation of two cells. Okay. So that two cells will again divide into form four cells. So it is simultaneous division means there won't be. Okay. You can see there will be formation of three cell stage. And if you can see your fifth page, this image are there. So this is tetrahedral stage, and this is isobilateral division, and this is decussate division. This is T-shaped. So you can see this one, T-shaped division. Okay, and this one is linear division. So here, so in successive division, what will happen? This separation of chromosomes will take place. They'll duplicate. After that, they'll be separated, and there will be formation of two cells. After that, so there will be diploid in nature. See, initially a cell will be in 2n condition. For example, if you take a human cell, there will be 23 pairs of chromosomes will be there. So in this first division, what will happen? It will duplicate. Duplicate means it will become 46 pairs. No, after 46 pairs, there will be equal division will have. Equal division often again 23 pairs there, 23 pairs here will have. Okay, comes. And next division, there won't be any duplication, but there will be separation. Then what will happen? 23 here, 23 here, 23 here, 23. Okay. So in here, 23 pairs. So this 23 pairs will be reduced into 23 only. Individual will be there. So this here, this 23 pairs will be there. From the female ovule, they will be containing 23 pairs. 23 cells. This 23 together, there will be formation of 23 pairs. Again, a diploid cell formation will be there. Okay. So in same as this same kind of process will happen in plants also. So this is simultaneous division of cytoplasm in microspore mother cells. Okay, here this is a microspore mother cell, and this is the two nuclei after meiosis 1. Okay, this is microspore mother cell after meiosis 2. Okay, so this is the tetrahedral tetrax, and these are the microspores. So different types of tetrads. So the tetrad types will be different types. There will be tetrahedral, so where pollen grains are arranged into different planes. Okay. So if you can see, if it is a 3D structure, you can see how they are arranged. So the pollen grains are arranged into different planes. 3 pollen grains are in one plane, and one lies centrally over the other 3. So here 1, 2, 3 are there. No, another one is here. So if you can see in 3D way, you can see it better. Okay. So next one is isobilateral. So in isobilateral, all the tetrad pollen grains are arranged in one plane. Okay? You can see all 4 cells now. Here you can see all 4 cells, right? Arranged in one plane. These 4 pollen grains visible in the same surface and arranged in 4 corners of the square. That is example is like a geomes. You can see this kind of microspore tetrads in Zea mays. Okay, and the T shape. This one T shaped is there, no? So in T shape, two microspores are arranged side by side, while the other two are one above one. So in this one arranged side, two are arranged side by side, and remaining two are one upon one are arranged. Okay. So for this example are Aristolochia species and Polyanthes species. Next one is linear. So in linear type, two microspores are arranged side by side. Sorry, in four microspores are arranged one above another. Right? You can see one above another. Four microspores are arranged. In the discated type, in this type, pollen grains are arranged in two pairs. One pair of microspores are right angle to other one. So you can see this. This will be like 90 degrees will be there. So one will be right angle to another one, another one will be right angle to another one. Okay. So these are the live images of Pollinia. So this one, next one is male gametophyte development. Okay. Here you can see it is starting. So this is the anther cross section. Within the anther, you can see the microsporocytes. Okay. They are initially in diploid stage. After meiosis one, they'll become two diploid cells. Okay, two haploid nucleus will be formed. After meiosis four, there will be four haploid nucleus will be there. Okay. After this, the tetrad formation will be there. So we have seen all five types of tetrad formation site. After this, microspore formation will be there. So whatever there will be cell wall formation between the four spores, and each will be individual after that. So within that, we have seen that germinative cell and nourishment, non-generative tissue. No? So this after pollen meiosis, we can see vegetative nucleus and generative nucleus, right? So this pollen meiosis too, it will see when they are, this is stigma. When they are attaching stigma, there will be formation of pollen tube. Okay. So in pollen tube, the sperm cells will be there and vegetative nucleus will be there. Here you can see. So generative nucleus is there, pollen tube, and this is nucleus of the pollen grain will be there. So they will travel into the female reproductive system, and one will within this within the pollen tube there will be division occurs. After that, from that they will get male two male gametes. One male gamete fuses with the ovule, so that is egg cell, and it forms an embryo. Another one fuses with the polar bodies to form a endosperm, 3 stage. So this is one is generative and this is vegetative. So here. So we have seen how this microspores are formed. Okay. So this microspores will be in 2n, one will be okay. So there will be generative cell as well as vegetative cell will be there. So when they are fusing with the egg cell, the one is forming the embryo, another one is forming the endosperm. So from the male anther, okay, there will be a microsporogenesis. From here, megasporogenesis will be, because that one is formation of egg. Okay. So this is a male reproductive organ and this is the female reproductive organ. So this pollen grains formed here will travel here. After traveling here, there will be formation of tube and then be fusing with this nucleus. So then you can see the formation of fruit.
Hello? Hello, sir.
Yeah, yeah. Okay. Now you can see this one, right? So you can see this a complete flower. So sepals will be there. Sepals means the greenish parts of the below the what flower will be there. Petals are colorful things. So this is a female reproductive structure that is completely known as carpel. So within the carpel, there are parts of bodies of carpel like stigma, style, and ovary will be there. So if you take this one as anther, so this one is stamen. So one is carpel and one is stamen. So with the stamen, if you take, there will be anther and there will be the tubular structure filament. Okay. From the pollen grains will travel from here to here and there will be a fertilization happens. So there will be different types of pollen grains will be there, like single and spike one, racine, carumbo, and umbel-like structures will be there. So different type of flowers will be there. So, so this is the hibiscus. Okay. So this is stigma. This is so these are the pollen grains here. So this is female reproductive organ. These are the male reproductive organs. So female reproductive organs will be top, so that there will be a chance of more chance of that cross fertilization occurs. But because of air or because of this honeybees will be there now butterflies, they'll come and stick. When they're sticking here, the pollen grains stick to their leg, and if they're standing here, so the fertilization take, takes place. So this is the microscopic view of the pollen grains. There will be different types of pollen grains will be there. So we'll go to this one. Next one is unit 2 is megasporogenesis and female gametophyte development. So this is the carpel. So carpel is again divided into stigma, style, ovary, and inside the ovary we can see the ovules, right? So the gynoecium represents the female reproductive part of a flower. So gynoecium may consist of a single pistil or carpel, those are known as monocarpellary. The gynoecium may consist of single will be mono not carpellary, that is hibiscus is the example for that. So the gynoecium may have more than one pistil, so that is multicarpellary. Xenocarpus or apocarpus. So this is stigma, and this is Stenocarpus ovary. More than one, the pistil may be fused together, Stenocarpus. So multipistillary Stenocarpus pistil of Papaver. Papaver carpus will be like this. This is mycelia. This one more than one. The pistils may be free, so they are free. So they are known as apocarpus, multicarpellary. Ovules are megasporangium. So marginal placementation, and this is marginal at one side. This is axial placementation. So the ovules are placed in the around the axis. This is parietal placementation. So they are at the borders, and this is precentral placementation. This is the basal placementation. Only one ovule present in wheat, paddy, and mango, and many ovules are present in papaya, watermelon, and orchids. So that is why if you can say wheat, paddy, rice, or mango. In mango, you will see only one seed, right? So because of the presence of one ovule, so only one seed formation will be there. And if you can see the papaya and watermelon or orchid, if you can see watermelon, in summer you'll be eating watermelon. So you can see there will be many seed formation because of each seed represents one ovule. So each ovule get fertilized with a microscope, that is pollen grain. So they'll be forming the seeds. Seeds are the used for next generation production of next generation plants. So the megasporangium is known as ovule. So this is the structure of ovule. So this is the so there are 6 types of ovules. One is orthotropous, and another one is anatropous, and another one is hemitropous. So this anatropous example will be like this one. So this is the embryo. This is the embryo sac, and this is the these are the polar nucleus, and this is the egg. This is the microcyte. Okay. So if you can see the arthrocarpus ovule, the body of the ovule is straight. The funicle, helium, and calaja and the micropyle lie in the straight line. So you can see here. So they all are here is the funicle, hilum, and micropyle. So they are in a single line. And this is anatropous ovule. Ana means backward or up. Tropos means turn. The body of the ovule becomes completely inverted around 180 degrees during the development, so that the micropyle lies very close to the hilum. The process of formation of megaspores from the megaspore mother cell is called as megasporogenesis. So this is a microscopic wave. So inside that, two nucleus will be there. In a majority of flowering plants, one of the megaspore is functional while the other one is degenerated. The functional megaspore develops into the female gametophyte, that is embryo sac. This method of embryo sac formation from a single megaspore is termed as a monosporic development. So development of female gametophytes will be like this. So this is the complete embryo sac, that is a female gametophyte. There will be collagen end. So there will be antipodals will be there. 3 will be there. So here polar nucleus will be there. That will be N plus N, 2N will be there. This is central cell. So this is egg cell. So with the egg cell only that the pollen grain nucleus will come and fertilize with this. That will form embryo. So these two are the synergids, and this is the piloform apparatus. Okay. The nucleus of the functional megaspore divides mitotically to form two nuclei. Each nuclei move to the opposite poles, forming the two nucleated embryo sac. So typical angiosperm embryo sac is at maturity. So the 8 nuclei is called 8 nucleated and 7 cell. Okay. So this is 1, 2, 3. 3, 4, 5, 6, 7, 8. Okay, 8 cells. This type of embryo sac is called polygonum type because it was derived by Strasburger in Polygonum plant. The function of the antipodal cells is to provide nourishment to the egg cell. The synergids act as a site of the discharge of the male gametes of the pollen tube. So here the function
So
of
what
this,
is synergids?
So these are the synergid cells where the they are cells. Okay, so they'll where the egg cells will be there. Sorry, the pollen cells will be there. No, pollen nucleus will come from there, and they'll fuse with this egg. So the synergids helps in the fusion of egg cell with the pollen cell. The synergids have special cellular thickening at the micropylar tip called piliform apparatus. We can say this one is the piliform apparatus on this synergids. So piliform apparatus play an important role in the guiding the pollen tube into the synergid. So this piliform tubes will be there. No? So the pollen tube will come from here. So they will release the pollen nuclei. So they will guide them to reach synergid. From the synergid to it will fuse with the egg cell. The central cell is the large central cell is as mentioned earlier, has a 2 polar nuclei. 6 of the 8 nuclei are surrounded by cell wall and are organized into cells. The remaining 2 nuclei called polar nuclei are situated below the egg apparatus in the large central cell. So this is what, this is a polar nuclei. This whole thing is known as central central cell. So this is the egg cell, and these two are the 2 polar nuclei. They are inside this central R. So you can see this clearly. Piliform apparatus. So they'll the pollen tube will come from here. They will release pollen nucleus here. They will transfer to here. So the pollination. Transfer of pollen grains to the stigma of a pistil is termed as pollination. Kinds of pollination: autogamy will be there, geonotogamy is there, gynogamy is there. Okay. So we are discussing this is a female, this one, stigma. So this is anther. From the anther, this pollen grains will transfer of pollen grains from the anther to the stigma. Okay, is known as the pollination. So this is autogamy. Autogamy means self-pollination will be there. So where self pollen grains will fall on the stigma. Autogamy requires synchrony in pollen release and stigma respectively. See, if it has to self-fertilize means, so development of pollen grains and development of ovules should happen same time. That is in a synchrony. That requires they should mature same time, so that the matured pollen grains will transfer onto the stigma. Then pollen tube formation will be there. They'll come and fuse with the egg cell. So the anther and stigma should lie close to each other, so that self pollination can occur. So see, they should be little closer. If not, the self pollination hard to occur. So another type is like, so casmogamas, flowers which are similar to flowers of other species with exposed anther and stigma. So this clistogamas are flowers which do not open at all. In such flowers, the anthers and stigma lie close to each other, so self-pollination will occurs. In clistogamas, underground flowers, so Camelina encephalis will be there. Camelina by bengalensis will be there. Camelina. So in that you can see the Camasmogamas. In Clistiogamus flowers, no chance of cross pollination because they are underground. So P flowers produce assured seed sets. Pollinators not required. Pollination agent not required. Advantages: 100% seed formation will be there. Disadvantages: offspring produced that are limited genetic variety. See, when self pollination is occurs, if you can treat this as a consensus marriage within the families, if they are getting marriage means there will be genetic similarities. If they are carrying any disease kind of thing, they will be carried. So there won't be much genetic diversity. Okay. So since they are already close, so there will be 100% what seed formation will be there, because since ovary and pollen grains are lying together, so there will be self pollination. So Geitonogamy, so transfer of pollen grains from the anther to stigma of another flower of the same plant. So if two hibiscus plants are close by close, if that if say that if you take a plant, so there will be many. I mean, there won't be a single flower. No? There will be two, three, four. Many flowers will be there. So if I in same as in this image, if you can see this flower is little above to this. So if this flower arise today and this flower is arise yesterday, if you can say, so this pollen development will be late than this one. No, so this pollen grains are coming and transferring into this means. So this kind of pollination is known as geotonogamy. Okay. So next one is geltonogamy. So the functionally cross pollination involving the pollen agents. Pollen agents like butterflies will be there, honeybees will be there. So because of pollen grains sticking to their legs and transferring onto the plant, another plant or say on the same plant and another flower, such kind of things are known as geotonogamy. Okay, so it is similarly to So xenogamy. Xenogamy, transfer of pollen grains from another anther to stigma of a different plant. So same two hibiscus plants are there. Two plants are there from one plant to another plant through this honeybees are known as xenogamy. So this is brings genetically different types of pollen grains of out to the stigma.
Sir, what is the difference between that xenogamy and geotonogamy?
So within geotonogamy within the same plant. Okay.
Okay.
Two different flowers of a same plant. Okay. So this is xenogamy is two different plants and the different flower. Obviously.
Okay.
Got it? So within
Yes,
this, great. Agents of pollination will be like butterflies will be there. Honeybees will be there. Some birds will be there. Okay. Even wind also sometimes cooperates in this transfer of pollen grains from pollination by wind. So you can see this paddy fields. So there is nowadays there is a male, female, different seeds plants they are planting. So they are taking a row for the two ends, and they're just simply traveling from one end to one end. So the pollination occurs. So simply fertilization means transfer of pollen grains under the stigma. So after this pollen grain, that will form a pollen tube. You can see this one, no? This tube. Can you see this arrow? Hand is you can see hand, no? So this pollen tube
yes, sir.
goes here. Goes here. Here we are talking about this, but this apparatus. Which apparatus? Yes. So they'll go and fuse with the egg cell, and another nucleus of this pollen grain will fuse with the polar nucleus that will form a endosperm, and this will form a embryo. Fertilization is the fusion of two dissimilar sexual reproductive units called gametes. Okay, so the process of by which two gametes fuse to become a zygote, which develops into a new organism that results in zygote is diploid. Parts of pollination, pollen tube will be there, progamy, calogamy, egg cells, female cells are zygote, endosperm, parthenocarpy, antipodal cells, and synergies. So pollen tube is a part of the male gametophyte of a seed plant. It acts as a conduit to transport the male gamete cells from the pollen grain, either from the stigma to the ovule at the base of the pistil. So here you can see this image. So this one is there, no, this green one. So the pollen grains are there. These green one lines are the pollen tubes. Okay. So on this, these are the red ones are orange ones are the pollen grains. So this green one, this bigger one is the stigma of the female organ. So this is the pollen tube. So that within the tube, this nucleus of the pollen grains will come and fuse with this one. So this one pollen tube, we have seen this. There will be which cell? The synergid cells will be there, no? So they'll come here. After that, they'll fuse with the egg. Porogamy. This is a commensate type of fertilization seen in angiosperms. Here, the entry of the pollen tube into the ovary takes place through the micropyle. This pollen tube is there, no? That is with touching with the embryo. So this is the female gametophyte. This is female polar nuclei. So this is egg nucleus. This is pollen tube. Okay, they are two fusing together. So this tube came inside. So after coming there, they both will fuse together. So from this tube, this two nucleus are there. No, blue color are the sperm nucleus. That is pollen grains. So this is the egg one. These two are the female polar nuclei. So one is fusing with the egg to form embryo, and another nucleus fusing with the polar nucleus to form endosperm. This is endosperm. This is embryo, and this full is endosperm. If you take coconut, no, so the edible portion is completely endosperm. So this we have seen. So this is under cross section and so microspore formation. So the forms or microspores, okay, will fuse with the egg inside the plant. So see, after fertilization, so actually this is known as double fertilization because one nucleus of the pollen grain fuses with the egg cell, and another one with fuses with the polar nucleus. That's why it is known as double fertilization. So this is the ultimately formation of seed. That seed will come, it can generate into the complete plant. Right? These are polar nuclei. So two nucleus inside one side. So this is endosperm is a tissue produced inside the seeds of the most flowering plants around the time of fertilization. Right? We have seen within the double fertilization. Second fertilization is the formation of endosperm. So you can say fertilization. So this pollen grains are releasing pollen onto. So this is anther. This is stamen. So this is stigma. So pollen grains are forming a stigma. So the tube formation is occurring. The form tube from the tube. So the sperm cells are two nucleus are transferring. One is fusing with egg cell and another one is fusing to polar nucleus. So that is forming endosperm. This is forming embryo. So you can see pollen tube enlarged view. So this will be two individual sperm cells will be there. So they'll go and go within the tube and fuse with the egg cell. So if you have any questions, you can ask. Actually, I just found your link. I joined in your group, so I'll share the notes there.
Okay, sir.
Sir, please provide some important questions also, sir. Very vast syllabus.
Important questions. Yeah, important questions. Since I joined in a group, no issue. So you can ask me the dots from in that group. Okay. I'll reply there. Even I'll share some notes. If it is on PDF or PPT form, I'll try to share the notes. So when is your exam?
From 7th June.
7th June, right? We have good time. So I'll try to share good notes. But I'll share PPTs now itself also. Okay.
So you're having floriculture notes?
Floriculture notes. Actually, I didn't see the syllabus. You just share the syllabus there. I'll make and I'll send it.
Okay. No, sir. In the fourth semester, we are having like two options we have to select. So I have selected from the botany only, biotechnology and floriculture.
Okay, if you have
So
chosen
if you are
biotechnology, if you
having,
have chosen floriculture,
yes.
no problem. I'll tell you that also. If needed, I'll make
Okay, sir.
some PPTs. I'll share to you. Because within this, I'm
Okay, sir.
dealing
Thank
with
you.
developmental biology and medicinal botany only.
Yes, yes,
So
yes. I know, sir. If you are having some notes,
no
so,
problem. See, because you are even a student of this thing, no, you may have many notes and all. I'll try with my knowledge. I'll try to make notes for you guys. You can share it on the group so that people will get benefit of it. Okay.
okay, sir.
So
Thank you,
this
sir.
is for
Thank
today.
you very much.
So we'll meet tomorrow. Tomorrow will be our last class, and we'll be closing that block two also. Right?
Okay, sir.
Right. Right.
Sir, block four also you are sharing the notes now, sir, from the block
Yeah.
four.
Yeah. Okay.
Okay,
Right.
sir. Thank you so much, sir.
Okay. Bye.
Yeah.